Hi friends, good morning, good afternoon, good evening. This is another edition of COVID-19 pandemic. Today I'm going to discuss about the role of antibodies in COVID-19 pandemic. Generally, our immune system produces antibodies to fight against viruses. An antibody is a protein that is capable of binding with high specificity to an antigen like virus. After the infection, one can become extremely sick if the immune system does not produce enough antibodies on time. The virus multiplies exponentially in the human system. So if the antibodies lag behind, then the virus becomes uncontrollable. The immune system takes few days to produce antibodies after the infection. Here I want to mention three important white blood cells, also called leukocytes, are an important part of human immune system, which are produced in our bone marrow. The first one is a macrophage or phagocyte, is a large white blood cell, also called monocytes. A macrophage has the ability to locate and eat particles of bacteria or virus or fungi or parasites. And also it triggers B cells to produce antibodies. Second one is T lymphocytes, or also called killer T cells. As we know, virus enters a cell and replicates within it. T cell is able to find and destroy virus infected cells that have been turned into virus making factories. T cell also triggers B cell to produce antibodies. Third one is B cell, also known as B lymphocytes, or another type of white blood cell. B cells fight bacteria and viruses by making Y-shaped protein called antibodies, which are specific to each pathogen and are able to lock onto the surface of an invading virus. It inactivates them and mark it for destruction by other immune cells. So what is the difference between T and B cells? The main difference between T and B cells is that T cell can analyze, only recognize viral antigen outside an infected cell and destroy them. Whereas B cells can recognize the surface antigen of viruses and produce antibodies to inactivate and destroy them. With any new viral infection, the body deploys macrophages which engulf the virus and breaks into pieces, and T cells which will find and kills infected cells with the virus. After about a week, the adaptive or acquired immune system kicks in, using B cell to make antibodies that can flag sick cells and viruses for more destruction even faster. Those antibodies stick around after an infection is over in case of a future invasion. So what is the role of antibodies in COVID-19 infection? The one is testing, which helps whether the patient or somebody has infection or not. The other one is in treatment. Antibody or serology testing for COVID-19 virus. Serology or antibody tests are blood-based tests that can be used to identify whether people have been exposed to COVID-19 virus. Serology-based tests analyze serum component of blood. The serum includes antibodies to specific components of COVID-19 called antigens or viruses. These antigens or viruses are recognized by the immune system as a foreign body and are targeted by the immune response. These types of tests are often used in viral infections to see if the patient has an immune response to a pathogen of interest, such as COVID-19 virus. These tests are looking for two antibody subclasses, IgM and IgG. IgM is the first that get produced in an immune response, but they are also relatively short-lived with a half-life of 5 to 6 days. So, detection of IgM against COVID-19 virus or its antigen indicates a recent or still ongoing infection. The IgG antibodies are more numerous in the end and may indicate that person has now immune to reinfection but don't know how long. One can realize then that if a person shows positive for IgM only, then they have well be recent or ongoing infection. And if they show only IgG, they may well have gone through an infection and could be immune to it. Showing both well, you are probably on the back end of infection, and showing neither could mean that you have not been exposed to virus at all. So then what is the role of antibodies in the treatment of COVID-19 infection? The first one is convalescent plasma therapy. Many options are being explored to treat COVID-19 infection. These include new drugs specifically designed to target COVID-19 virus, as well as existing drugs designed to treat other diseases. By far the oldest treatment being tested is convalescent plasma therapy. This involves using plasma from people who have recently recovered from COVID-19 infection and infusing it into patients who currently have the disease. Plasma is the liquid portion of the blood that remains when all red, white blood cells and platelets have been removed. Plasma therapy is 100-year-old method. Many hospitals all over the world are collecting blood from patients recovered from COVID-19 infection, so the plasma in it can be directly transfused into the patients with a severe disease. The use of such convalescent plasma was tried as early as January as an emergency treatment in China for COVID-19 infection with promising results. That is not an ideal solution because plasma, which has a large mixture of antibodies present in small amounts. Using convalescent plasma involves the transfer of antibodies from donors who have already mounted an immune response, thus offering immediate some protection to the recipient. But the most important is treatment of COVID-19 infection with monoclonal antibodies. Generally, blood-derived antibodies are termed polyclonal antibodies because they are not derived from a single B cell or its progeny, but are instead a product of many different B cells in the blood, binding in these slightly different ways to the same antigen. As a result, the method yields a very impure product that contains wide variety of antibodies. To avoid problems associated with use of such antibody mixtures, scientists developed the technology to produce monoclonal antibodies. A monoclonal antibody is an antibody that is a product of a single B cell clone. Production of monoclonal antibodies in the lab involves combination of two types of cells. The first type cell is a B cell that is produced a single unique antibody. The second type of cell is a myeloma cell. The specific antibody producing B cell is fused with non-producing myeloma cell to form a hybrid cell called hybridoma. This hybridoma can produce the desired specific antibody in a high concentration and in pure form. As a result of this development of monoclonal antibody technology, scientists can produce large amount of pure and highly specific antibodies. These are like guided missiles which would hit the virus. But mass production of monoclonal antibodies is difficult and they are awfully expensive. In a lab setting, monoclonal antibodies are produced by introducing spike protein of COVID-19 to a mouse and then fusing B cells from the mouse's spleen to myeloma cells. The resulting hybridoma cells are cultured and will produce monoclonal antibodies against COVID-19 virus. Separate studies in Israel and Netherlands claims to have created monoclonal antibodies that can block or neutralize COVID-19 virus, a potential future treatment until a vaccine becomes available. Both efforts, which are in their initial stages, hope to eventually treat or prevent the development of COVID-19 infection and may stall the spread of the pandemic. This is certainly a very promising discovery coming from robust scientific approach and should be viewed as a reason for optimism. At the moment, there is no definitive treatment or vaccine for COVID-19 pandemic, which has killed more than 318,000 people worldwide and infected more than 4.8 million. In the meantime, we will continue to follow the guidelines set by our local authorities. Frequent hand washing, staying at home, and maintain social distancing with wearing face mask in the public areas. We are all in this together and we all have a role to play. Till then, take care, stay safe. Thank you for watching this video.